Exocytosis is a means by which substances are released from cells, molecules, signaling molecules. In our previous video, we discussed gated channels, which is a way to allow access for things to enter cells and exit cells. And exocytosis is another means, along with something known as endocytosis, which, as we're looking at this video, keep in mind endocytosis is the exact opposite. That is to say, exocytosis is releasing contents from cells. Endocytosis allows for contents to be brought up into cells. I'm Matt Halter. It's Corona Summer 2020, and this is Human Physiology. Okay, looking at this slide right here, we see a big cell in blue, and this red thing is what's known as the secretory vesicle, and that will contain some sort of molecule, chemical signaling molecule. For example, insulin is released from secretory vesicles from regions of the pancreas, and we'll talk about that in detail in subsequent videos. The secretory vesicles can also contain channels in them, and they get embedded within this cell membrane via exocytosis. So we're going to talk about all of that in detail. So once again, here's a secretory vesicle, and we see some sort of molecule in here. At this point, I don't really care what this is. Like I said in the previous slide, this could be insulin. This is how insulin is released via exocytosis. So these green things are molecules that are confined within the secretory vesicle until they are stimulated to be released. And that stimulus, oftentimes, not always, is calcium. Calcium, the influx of calcium into cells will facilitate the process of exocytosis, which ultimately will release these green molecules out to the extracellular fluid. Keep in mind, everything within the cell is the intracellular fluid. Everything out of this cell is the extracellular fluid. Here we can see calcium has pushed that secretory vesicle to the periphery, right next to the plasma membrane, where it is going to begin to fuse with the plasma membrane. So as it fuses with the plasma membrane, it starts to release the contents of the secretory vesicle into the extracellular fluid. If this was insulin, that insulin would leave the cell, move into the extracellular fluid, specifically the interstitial fluid, and then proceed immediately into the blood plasma. That is the basis of exocytosis. It is the release of intracellular contents confined within secretory vesicles, which then get released out of the cell via the process of exocytosis. And there we see the complete fusion of the membrane from the secretory vesicle is actually fused with the membrane of the actual cell, of the plasma membrane. Here is what I was alluding to at the beginning of this video, where secretory vesicles can contain channels, channel proteins. So what I had been talking about are molecules that get released in the extracellular fluid. The goal with exocytosis in this case is to put in place these channel proteins, which we see around the membrane of the secretory vesicles, to put these in place within the plasma membrane of the overall cell. So if we can get this red membrane of the secretory vesicle to fuse over here with the cell membrane, then we will put channels in place that things can move through those. So I've skipped a few steps. There has to be some sort of stimulus. In the previous example, the stimulus I showed you was calcium. So this could be, a stimulus could be antidiuretic hormone. So if these channels right here, which you see are now embedded within the overall phospholipid bilayer of the cell, These would be known as aquaporins, which would allow water to move into this cell. We see this within the nephrons, which is the structural unit of the kidney, which we'll talk about in detail in a later video. But it is the hormone antidiuretic hormone, or otherwise known as ADH, which facilitates the process of exocytosis, placing these water channels, otherwise known as aquaporins, into the cell membrane. Insulin, which I discussed earlier, also causes this to happen with glucose channels to allow for glucose uptake within cells. So please don't confuse this with me describing the release of insulin. This is the other end of that spectrum, where once insulin is in the bloodstream, it will allow for glucose uptake within cells by moving these glucose channels into the phospholipid bilayer of this cell. That is it for exocytosis. Endocytosis is the exact opposite. It brings entities, molecules into cells. Enjoy your day.